project of biology. Today we're going to be talking about recombinant DNA. What is the meaning of recombinant DNA? Recombinant DNA, molecules of DNA from two different species that are inserted into a host organism to produce new genetic combinations that are the value to science, medicine, agriculture, and industry. Since the focus of all genetics is the gene, and the fundamental goal of laboratory genetics is to isolate, characterize, and manipulate genes. Although it is relatively easy to isolate a sample of DNA from a collection of cells, finding a specific gene with DNA sample can be compared to finding a needle in a haystack. Consider the fact that each human cell contains approximately 2 meters, which are 6 feet, of DNA. Therefore, a small tissue sample will contain many kilometers of DNA. However, recombinant DNA technology has made it possible to isolate one gene or any other segment of DNA, enabling researchers to determine its nucleotide sequence, study its transcripts, mutate its highly specific ways, and reinsert the modified sequence into a living organism. Now we're going to be discussing the steps of making recombinant DNA. What are the steps that make the recombinant DNA? First, there are 5 steps. The first one is identification and isolation of the gene of interest. First of all, from where do we get the gene of interest? We get it from the genomic library, which is cDNA. Now for step number 2, we have to insertion of the isolated gene into a vector. What is a vector? A vector is any DNA molecule that has the ability to replicate inside the host to which the desired gene has integrated for cloning. Now for step number 3. Now first, this diagram it illustrates step number 2. Here we have the DNA sequence of interest, and restriction enzymes cut out the DNA of interest and opens the vector. And the DNA of interest opens the vector, and DNA ligase joins the two DNA sequence together. Vector containing DNA of interest, as you can see in the steps and the arrows where they point to each step. Now for step number 3. We had in the introduction of the host into a suitable host. Introduction of the vector into a suitable host. There are 2 types, physical and chemical transfer methods. As you can see this illustration, this is how it should be. This process is called transformation. Now moving on to step number 4, selection of the transformed host cell. After step 3, we will be getting 3 types of colonies. The first one is non-transformed bacterial cell. The second one is transformed bacterial cell with altered vector. The third one is transformed bacterial cell with recombinant vector. As you can see, these are the 3 types, and they are shown in the 3 photos that they appear in front of you. Step number 5 is multiplication expression of the introduced gene is in the host. Finally, after step number 5 is done, we are done with all the steps. Now we have to consider something. There are uses and aims of recombinant DNA. Why do we need to have recombinant DNA, and why does it take all of these steps to be done, and what is the aim of doing all of that, and the use of it? Recombinant DNA technology has also proven important to the production of vaccines and protein therapies, such as human insulin, interferon, and human growth hormone. Okay, so this is very important. It is also used to produce clotting factors for treating hemophilia and in the development of gene therapy. So after all of these slides, we should understand that recombinant DNA is very important, and it has very specific steps. And we should have understood what the meaning of recombinant DNA, and that's